Hello and welcome everyone to the 12th webinar in Bio-Rad's webinar series on Droplet Digital PCR. My name is Carolyn Reifsnyder and I'm the Global Marketing Manager here at Bio-Rad's Digital Biology Center and your host for today's webinar. First, just a few housekeeping notes on the webinar logistics. The webinar will last approximately 1 hour. Dr. Jennifer Berman will spend about 45 minutes discussing the genome editing revolution and how tools like TALENs and the CRISPR-Cas9 system are advancing our ability to edit genomes of any cell. Droplet Digital PCR is emerging as a powerful tool to accelerate optimization of genome editing protocols through its ability to detect edited alleles present at frequencies lower than 0.5% with a rapid, low-cost readout. Following the presentation, we will have 15 minutes of live Q&A with Jen. The webinar will be available on demand after our live presentation today, and you'll be able to download the slides from today's talk, along with the most recent publication list, system brochure, and useful web links from the resource list on your screen. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen to submit your questions directly to us. We'll get to as many of your questions as we have time for today. However, if we can't get to your question, we'll personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now I'd like to introduce our speaker, Dr. Jennifer Berman. Jen is a staff scientist here at the Bio-Rad Digital Biology Center, where she leads advanced applications for Droplet Digital PCR and assay development projects in cancer and applied genomics. She earned her PhD in genetics at the University of California, San Francisco, specializing in the genetic basis of aging and lifespan as a Howard Hughes Medical Institute predoctoral fellow. Following her postdoctoral work at Stanford, Jen is focused on the contribution of human genetic variation to health and disease in the personal genomic space. The title of Jen's talk this morning is Ultra-Sensitive Quantification of Genome Editing Events Using Droplet Digital PCR. Jen, over to you.
Thank you for that introduction. So my role at the Digital Biology Center is to connect our leading-edge technology, Droplet Digital PCR, with leading-edge biology applications. Today I'll give a brief introduction to ddPCR for those new to the system, but I encourage you to refer to other webinars in our series for more detailed background on the technology. I will then give an introduction to the genome editing field, and I will introduce how we can use ddPCR plus some interesting assay strategies to detect and quantify rare edited alleles. Cutting-edge research is increasingly the science of rare species that are difficult to detect and quantify, or small fold changes that are difficult to discriminate. One example is circulating cell-free DNA, for instance from a fetus or a tumor, present at maybe only 1,000 to 5,000 copies per mil of plasma. Studies of tumor heterogeneity might be looking at mutations or copy number changes present in only a small fraction of cells. Now, intercepting and assigning meaning to these rare events will drive the future of medicine. It's treatment that is non-invasive and personalized to a patient's genetic profile. Examples include the use of patient-derived stem cells for personalized therapies, as well as the oft-talked-about liquid biopsy, where cell-free DNA from plasma or other sources might be used to track a tumor. Droplet Digital PCR, or ddPCR, using the QX200 system enables highly precise and sensitive nucleic acid quantification. The workflow is simple. A 20 microliter PCR reaction, including your sample, primers and probe, and droplet-compatible master mix, is converted using the droplet generator or AutoDG to make 20,000 nanoliter-sized droplets. Droplets are transferred to a 96-well plate and thermal cycled to endpoint. Now, droplets containing the target become fluorescent and are counted positive by the droplet reader. Droplets without target are counted negative. So one reaction effectively goes from analog to thousands of discrete yes or no answers. Oops, sorry about that. The key technical advantages of ddPCR include the following. First, absolute quantification. There is no standard curve, and we have a readout in copies per microliter. And because we read at endpoint, there is less worry about PCR efficiency and increased resistance to inhibitors. The high precision and reproducibility allows the ability to discriminate small differences in gene copies. Third, high sensitivity enables detection of rare events, for instance, cancer mutations, virus, and relevant to today's talk, genome editing events. This cartoon illustrates how high sensitivity is enabled by sample partitioning. Droplets increase the relative abundance of rare species by titrating out high copy backgrounds. In this example, the bulk sample has a mutant species present at 1 in 1,000 copies of background molecules. 40 mutant target molecules are present in a background of about 40,000 wild-type copies. When the sample is partitioned into 20,000 droplets, 40 droplets will contain the mutant target, now at a 1:3 ratio to wild-type. So what is happening here is that droplet partitioning effectively increases the signal-to-noise ratio of a rare target. This high sensitivity is particularly relevant to detection of rare cancer mutations, for example, in circulating cell-free DNA, as I mentioned earlier. This slide shows validation data from one of our most popular mutation detection assays, BRAF V600E. Now, activating mutations in BRAF are common in late-stage melanoma, and V600E substitution represents more than 80% of these mutations. For our mutation detection assays, we use a single primer pair to amplify the mutation site and 2 competitive probes to detect mutant or wild-type copies. Mutant is in FAM and wild-type in HEX. The 2-dimensional droplet fluorescence amplitude plot shows a 0.1% mutant well, displaying each droplet in the reaction. The no template droplets are in gray, the wild-type single positives are in green, the mutant single positives are in blue, and the mutant and wild-type double positives are in orange. The limit of detection plot below shows 0.1% mutant detection in a single well, or roughly 1 in 1,000, with clear separation from the wild-type only negative control. This technical capability of ddPCR has enabled many cancer researchers across a wide application space, including rare mutation and cell detection, detecting somatic copy numbers, alterations, and microRNA detection. I encourage you to watch one of our most recent webinars on ddPCR cancer applications to learn more. So what can an ultra-sensitive molecular counting instrument do for the genome editing field? I'll spend the rest of this webinar on genome editing and how ddPCR is becoming an ideal companion technology for researchers investigating and implementing genome editing protocols. Genome editing, or engineering, simply refers to the precise sequence-specific modification of a genome. In some cases, this system can act essentially as a search and replace function. Genome editing is an increasingly prevalent part of the molecular biologist toolkit and is actively being developed for therapeutic indications. So why is genome editing so hot today? While fundamental editing strategies have been around for a while, the recent and exciting genome editing craze is the result of an expanded, improving editing toolkit, in particular with the advent of the CRISPR-Cas9 system, which I will talk more about. Editing today is easy, efficient, and affordable, and can even enable inducible or transient regulation of gene activity. With these improvements, gene editing is becoming a go-to technique to address unmet need for genomic manipulation in basic research and medicine. Genome editing has already proven to be a powerful strategy for basic research and provides new ways to do drug and disease studies. The system works in all model organisms tested and can be used to create transgenic mice in a matter of weeks. Genome-scale functional screening in human cells has already been performed by multiple groups using lentiviral vectors to systematically deliver CRISPR editing components with paired guide RNAs. And researchers are leveraging genome editing to create human cellular disease models, even based off patient-derived induced pluripotent stem cells. This powerful approach optimizes disease-in-a-dish type studies through generation of perfectly isogenic controls, helping to minimize confounding variables. Finally, CRISPR interference, or CRISPR-I, and other approaches are allowing precise modulation of gene activity through the recruitment of transcription and epigenetic regulators to specific DNA sequences. The editing community has been quick to recognize the potential for genome editing in diagnostic and clinical applications. Genetic sequence information of patients is increasingly available and is already used in cancer and other fields for diagnostics and for predicting response to therapy. This first panel presents a scenario where patients get sequenced and their personal variants of unknown significance are functionally interrogated through genome-edited cell lines. For example, one might be able to ask if that patient-specific mutation confers resistance to a drug's mechanism of action. The second panel highlights a recent report that used editing in HIV patient-derived T cells to disrupt function of CCR5, an essential co-receptor for HIV. These patient-specific HIV-resistant T cells were reintroduced into the HIV patients with encouraging preliminary results. Editing cells in the hematopoietic lineage, whether for blood diseases like sickle cell anemia or for immunotherapy, is at the forefront of a field known as therapeutic editing. The main methods for genome editing are shown on the following slides. Other methods, such as meganucleases, are also used but will not be the focus of this talk. Zinc finger nucleases, or ZFNs, and transcription activator-like effector nucleases, or TALENs, are similar in that they bring the endonuclease domain of FOC1 to a specific site in the genome through fusion to sequence-specific engineered DNA binding domains. For TALENs, sequence recognition is encoded by tandem arrays of 33 to 35 amino acid repeats. Specificity of ZFNs and TALENs is enhanced through the use of dual adjacent half sites. TALENs are more extensively used today over ZFNs due to comparatively high activity, better design density, and open source nature of reagents. The CRISPR-Cas9 stands for Clustered Regularly Interspaced Short Palindromic Repeat CRISPR-Associated Nuclease 9 System. CRISPR is an adaptation of a bacterial adaptive immunity strategy. In this editing system, short sequence-specific guide RNAs target the Cas9 nuclease to desired editing sites. Guide RNAs are now commonly engineered to combine necessary functional components with the sequence-specific regions. While off-targets were a concern with the first CRISPR systems, the use of paired mutant Cas9 nicases can improve specificity by more than 100-fold. These nicases include the D10A and H840A variants, which nick their target sites, as well as catalytically dead Cas9, or dCas9, fused to a FOC1 endonuclease domain. By putting the modularity at the level of the nucleic acid, CRISPR has made genome editing a fast and reliable part of any molecular biologist's toolkit. In this slide, I will briefly highlight the advantages and limitations of these three popular editing systems. I've circled some key pros in green and cons in red. CRISPRs can be easy to create, since unlike ZFNs and TALENs, no protein engineering is required. CRISPR editing is efficient, although as I mentioned, off-target effects must be monitored. TALENs are also extensively used and are highly efficient, but some sites, such as those rich in methylation, are intractable. And ZFNs have been historically important, but their lower success rates and potential for cytotoxicity are known technical hurdles. Okay, so how does genome editing work? Well, first, the selected nuclease, delivered as protein, RNA, or DNA, and associated reagents like donor molecules and guide RNAs, are delivered to cells using transfection or viral transduction. The site-specific nuclease induces double-strand breaks at the sequence of interest. This, in turn, induces activity of the routine endogenous repair mechanisms present in the cell. If a donor molecule is present, including homology arms, homologous recombination can occur, and via homology-directed repair, or HDR, can introduce a point mutation, correction, or even knock in larger sequences, such as a new promoter or GFP tag. A different repair strategy is nonhomologous end joining, or Nhej. This is shown towards the right. This is an error-prone repair system, and basically slaps broken DNA back together, and in so doing often introduces small insertions or deletions, or indels. Nhej can thus result in a frameshift mutation and knock out gene function. But when you are trying to introduce a specific edit via HDR, Nhej is not desirable and can represent collateral damage. What I'll introduce today is that ddPCR can be used to rapidly and sensitively detect these rare edited alleles generated by both HDR and by Nhej. And I will show data from real edited samples. A key challenge in genome editing is simply that edited alleles can be rare and hard to detect. While edits are site-specific, successful modification of targeted sequence, in particular when knocking in larger sequences, might occur in less than 5% of exposed genomes. This is highly locus and cell type dependent. For instance, editing in induced pluripotent stem cells or primary cells, shown here, might occur in less than 1% of genomes treated with editing reagents. In addition, Nhej is often more prevalent than HDR. Now, current strategies for detection of rare edits, like gel-based methods or Sanger, are not sensitive enough to catch these, or require time-consuming isolation and expansion of single clones. NGS is possible, but introduces cost and time, and in some cases bias against longer edits. So genome editing is full of promise, but key technology development challenges exist and need to be overcome in order to maximize its potential. We need to detect these rare edited alleles in a rapid, cost-effective, high-throughput manner to allow for quick iteration of protocols. There is a need for empirical validation of guide RNA efficiency. How do we connect the computational predictions to in vivo performance? Optimizing the HDR to Nhej ratio will likely need to be solved on a locus and cell type specific basis, and is especially critical for therapeutic editing. And off-target effects need to be minimized. ddPCR offers a powerful tool, however, for empirically and experimentally addressing these hurdles. So I'll go into some examples here. In this 2014 Nature Methods paper, researchers from Bruce Conklin's lab at the Gladstone Institutes applied ddPCR for sensitive detection of genome editing events in induced pluripotent stem cells, or iPSCs. Their lab wanted to introduce specific mutations in iPSCs to study how these mutations contribute to cardiomyopathies. They used both TALENs and CRISPR methods to generate mutations in 5 different genes important for heart cell differentiation. In their workflow, point mutations were introduced using an oligonucleotide donor. To detect rare point mutant edits, they used an allelic discrimination TACMAN assay in ddPCR. The researchers not only knew that iPSC editing events are rare, but they also wanted to express low levels of nuclease in order to minimize off-target effects. So they wanted to see if ddPCR could detect and quantify these rare events and allow for an improved protocol. The research introduced a new strategy, actually borrowed from yeast genetics, of mutagenesis followed by Cid selection for edited iPSCs. In this workflow, instead of cloning out cells from an edited pool, then growing them up to screen for edited clones, or using antibiotic selection, the idea is to use ddPCR as a sensitive tool to screen pools of cells, and once identify, expand those pools to enrich for the edited cell. For this approach, they were able to use ddPCR to detect editing events as rare as 1 in 5,000. In practical matters, this reduced by tenfold the active work time needed to screen for mutant clones. With older methods to get something this rare, they needed to screen 2,000 clones. Here, they only screened 11. And they are now routinely using this method to generate dozens of independent iPSC clones for their heart stem cell studies. Okay, so 2 assay strategies for detection of introduced edits are illustrated here. For small HDR edits, such as a point mutation in the paper I just discussed, an allelic discrimination assay with competitive wild-type and HDR probes is appropriate. One primer needs to sit outside the donor molecule, as shown in this schematic, in order to quantify integrated events. Some HDR edits aim to introduce larger sequences, such as a fluorescent tag or new promoter. One assay strategy for larger edits is represented in the bottom panel. The target assay employs a primer and probe inside the exogenous sequence, with one primer outside the donor region. A second reference assay can be duplexed or run in a second well to count wild-type copies. In this case, it is advisable to use an assay whose amplicon length is roughly the same as the target edit assay. Okay, so in particular, for large edits, a long homology arm might be necessary for effective edit introduction. Since one assay primer needs to sit in the targeted genome outside of the donor sequences, this can mean some long amplicons. This data shows how increasing the extension time while using a 3-step thermal cycling protocol can be useful in maximizing efficient amplification. The template shown here is a plasmid being quantified with an assay of increasing amplicon length. By adding a discrete 2-minute extension step, amplicons longer than 1 kb were amplified. Some thermal cycling rules of thumb for long amplicons are listed below. Okay, so on to another type of edit. We're also developing assay strategies for the detection of Nhej, and thus providing a useful readout of nuclease activity. The challenge with Nhej is that Nhej produces a heterogeneous population of insertion and/or deletion alleles. This is illustrated in this figure, where sequencing results are aligned to a site targeted by Cas9 nucleases. There are 2 predicted cut sites, which are indicated by the red triangles. Note the deletions seen in this sample are of variable length and often asymmetric. So how do we use an assay to detect these things? To quantify Nhej, we are using a strategy we call the drop-off assay, but this is a design previously applied in cancer research. This assay consists of a single amplicon that spans the targeted cut site. 2 probes are used. A FAM-labeled reference probe counts all copies and sits away from the cut site. A hex-labeled Nhej, or so-called drop-off probe, is complementary to wild-type sequences and is positioned directly on top of the predicted cut site. For example, 3 base pairs upstream of the PAM-NGG sequence for CRISPR. So looking below, on a wild-type molecule, both probes bind, causing droplet fluorescence in both the FAM and hex channel, seen as a cluster on the 45-degree angle. That's at the top right of this slide. On an Nhej allele, for example containing a deletion, the drop-off probe fails to bind, resulting in the loss of hex signal. Droplets containing only Nhej alleles thus appear as a separate FAM-only cluster, separable from the wild-type droplet group. And because we can cluster this, we can quantify it. In this example, our assay is being run on wild-type DNA combined with a synthetic double-stranded DNA template at 1%. And this template contains a 1 base pair deletion. The drop-off assay is unique in ddPCR because it is a 3-cluster assay. So I want to spend a moment explaining proper analysis. As I have previously shown, the 2-dimensional plot contains 3 clusters: empty droplets with no template, Nhej single positive FAM droplets, and a wild-type cluster that is both FAM positive and hex positive. Note that as loading and/or Nhej fractional abundance increases, seen here from left to right, a double positive tail appears off the wild-type droplet group. We infer these droplets to contain both wild-type and Nhej template. However, since they don't cleanly separate from the single positive clusters, we draw a threshold to include these droplets with the wild-type group. Nhej quantification is then assessed using only the Nhej single positives. We are releasing an analysis tool to ensure proper quantification based on this new assay strategy. Okay. We validated this assay strategy on wild-type DNA containing a serial dilution of synthetic double-stranded DNA template representing a 1 base pair deletion at the cut site. Validation data, shown below, shows the sensitivity of this assay down to about 0.1% for editing the progranulin locus, which is important in frontotemporal dementia. This sensitivity far exceeds what is possible with gel and Sanger-based methods. It also provides 1 readout of nuclease activity and guide RNA efficiency. I'd like to just mention that similar performance is observed when we've done this with a 1 base pair insertion positive control. On the right, we have example 2-dimensional droplet plots showing real edited samples, one with very little Nhej and one with lots of Nhej below. I'd like to mention that these edited samples on this and subsequent slides were generously provided by our collaborators, Ichi Miyaoka and colleagues in the Conklin lab. We use this assay strategy to quantify Nhej in a series of CRISPR-edited HEC293T cells. Edits present in less than 1% of the alleles could be detected and discriminated from the negative control. One sample was serially diluted, and wild-type and Nhej alleles quantified. Excellent linearity is shown in this experiment. One application for this assay is quantitative assessment of guide RNA usage. In this experiment, we are looking at cells edited with a paired Cas9 nickase. This means that 2 guide RNAs were used to create the double-stranded breaks. And each of these cut sites could potentially be a nexus for Nhej generation. 3 drop-off assays are shown here. With the nickase assay, 2 Nhej probes, named A and B, each cover one of the 2 nick sites. The CRISPR-A assay only contains 1 Nhej probe for the first nick site. The CRISPR-B assay contains one Nhej probe, this time sitting on the second nick site. The 2D plots on the right qualitatively show how in the same edited sample, the CRISPR-B site contributes more to the total Nhej count than the A site. You can see this in the density of the blue Nhej cluster. So we can quantify this effect. I'm now showing 2 loci here, RBM20 and progranulin. The blue bars are Nhej quantification, shown in copies per microliter of ddPCR. We have one sample assayed 3 ways, shown in each case that all or the majority of Nhej is coming from one of the 2 nick sites, in each case the CRISPR-B site. The wild-type graph on the right shows that sample loading is equivalent, important for these comparative purposes. There's at least 2 hypotheses that could explain this. Either one guide RNA is more efficient than the other, or one sequence is more prone to Nhej than the other, or maybe some of each. This data demonstrates comparative measurement of guide RNA efficiency and an empirical validation of cut site efficiency. Okay. I'd finally like to introduce an advanced assay strategy we've been working on in development with our collaborators in Bruce Conklin's lab. So, as I mentioned earlier, even when precise HDR edits are intended, Nhej can also be the result of genome editing. This has even been observed in a single cell, where HDR was introduced on one allele, and upon sequencing, the researchers find that Nhej was found to have disrupted the other allele. And this is simply not a scenario that's acceptable, in particular, if you want to use these edited cells in therapeutic indications. So we simply asked whether we can combine both HDR point mutation edit detection with Nhej detection in a single well, essentially combining the point mutation assay with the drop-off assay. This so-called total edit assay would then allow for simultaneous HDR and Nhej allele quantification in a single well. Okay, so a total edit assay schematic is shown here. For this assay, we have one amplicon, about 150 base pairs in length, that spans the edit and predicted cut site. A FAM-labeled reference probe detects all copies and is designed distant from the cut site. A second FAM-labeled probe is an HDR mutation detection probe, able to bind the desired point edit. A single hex Nhej probe sits directly over the predicted cut site, and its loss detects the presence of an Nhej indel. Below, the 3 allele types are represented in 2D droplet space. The wild-type well is on the left, and a positive control well containing both Nhej and HDR synthetic alleles is on the right. Now, compared to wild-type or Nhej droplets, containing HDR alleles experience a further gain in FAM fluorescence, which you can observe in that orange cluster labeled HDR. So with this type of assay, we're able to distinguish and do quantification of NHEJ and HDR alleles in a single well. This shows application of a total edit assay to a TALEN-edited real sample, ATP7B R778L, which is important for Wilson's disease. For wild type as well as edited HDR and NHEJ alleles, we report abundance in copies per microliter. So what we have here is linear quantification over a two-fold dilution series of this sample, with as little as 5 nanograms of sample input. With absolute quantification of HDR and NHEJ edited alleles, the Conklin Lab is evaluating what editing strategies work best for the loci they study. So this graph shows pilot data using this assay, illustrating NHEJ and HDR fractional abundance by ddPCR in HEC293T cells, edited at the RBM20 locus using different editing tools. So here we are able to sensitively detect edited alleles, and furthermore, this method lends itself to evaluation of strategies that yield more HDR than NHEJ. So this assay thus provides a powerful readout for optimizing editing protocols. Okay, so in conclusion, I've introduced how ddPCR can be used as a powerful companion technology for genome editing workflows. The ability to rapidly and sensitively quantify NHEJ and HDR alleles in a high throughput and cost-effective manner provides an important readout for genome editing optimization. We are continuing to develop new ddPCR assay strategies to quantify the vast diversity of edits desired by the genome editing community. And we have helpful tools and documents coming out, for instance, an applications note coming out very soon. So I'd like to thank you for your attention today. Our slides include some references and suggested reading for you to come back to if you're interested. And with that, I'm going to turn it back over to Carolyn for the questions session. Thank you so much.
Jen, thank you for providing us with the exciting overview of genome editing in Droplet Digital PCR. Before we open up the line to questions, as a quick reminder to our attendees, today's webinar will be available on demand within 24 hours. You can download the slides, including those references and suggested reading from what Jen just mentioned in her talk, along with the most recent publication list from the resource list on your screen. Additional webinars in the series are also available on demand for your reference, as Jen mentioned. So now we're going to go ahead and take some questions from our audience. So Jen, the first question comes, if I'm already detecting an HDR edit with a Droplet Digital PCR assay, can I go back and quantify the NHEJ?
Okay, great. Yeah, that's a really good question. So the assay strategy I showed at the end has both HDR and NHEJ detection in one well, but it is important to make the point that you can actually make one sample prep and split it into 2 wells and use the drop-off assay to quantify NHEJ in a well, and mutation detection assay to do detecting the point mutant in another well. So what you could do in that case, if you're already evaluating with, for instance, like the allelic discrimination assay, is you can go back and look at your sample with simply a drop-off assay. So the total edit assay is very cool, but you can also dissect the 2 components with 2 assays in 2 wells.
Excellent, thank you. The next question, actually, we have 2 audience members asking the same question, so it must be popular. How can the Droplet Digital PCR technique be used, if possible, for off-target gene editing?
That's a really good question and also a very hot topic these days. So how do you use this technology to detect off-targets? So we're developing some assay strategies around that, but one clever idea would be to simply use the drop-off assay, so the Nhej Quantification assay. And what you might do is, for instance, if you're using certain computational tools for designing your TALENs or your guide RNAs, you can computationally identify, let's say, the top predicted off-targets. And if you design a drop-off assay to those predicted sites, you can then directly quantify Nhej at those off-targets. So sort of combining, because I know when you're designing your TALENs or your CRISPRs, you're often using these tools to minimize off-targets, but you get some predictions. So you could just go ahead and use an assay designed to those predicted off-target sites and begin to understand the off-target landscape for your editing tools.
Great, thank you. So the results that you showed throughout your talk, have they been validated by sequencing or another approach?
Yeah, so that's also a really good question. So how are we validating this? We're working on that right now. We're going to be looking at sequencing. So it's interesting because a lot of people come to us because, for instance, gel-based techniques have failed them or the qPCR hasn't worked to their satisfaction. So how you validate digital, you don't want to take a step back necessarily and use a gel-based system. So we are working on evaluating this orthogonally using sequencing.
Okay, thank you. Next question, actually common question coming in from the audience here. How do I design, or do you have any specific tips for designing these types of assays?
Yeah, that's a really good question. So we are working, we are compiling our design guidelines. We already basically have guidelines for the HDR type assays because those are more or less akin to mutation detection assays in cancer, with the modification I mentioned of you want to make sure one of those primers sits outside the donor molecule. But for the drop-off assay, we are compiling our assay design guidelines, and we're also working on developing tools to do more on-demand assays, but that's in development. So we're working on solutions for our customers.
Okay, excellent. Next question is it possible to use Cyber or Evergreen?
Yeah, that's a really good question too. So everything I've shown here today was TACMAN-based. But there are possibilities. So, for example, if you're trying to create a deletion, we've seen people using Evergreen to do this. So our system does Evergreen, and if you're, for instance, creating a very defined deletion, Evergreen could be an appropriate way to do that. So we're working on those types of strategies as well.
Okay, excellent. We do have a question from the audience to talk a little bit more about the Cid Selection Strategy. I'm not sure if that's something that you would be able to speak on or not, but during your talk you had mentioned that some of the groups are using a Cid Selection Strategy for genome editing.
Yes, so is there a question or just to discuss the strategy? So on the slide I showed, it basically shows the idea. So you might be performing your mutagenesis, in this case genome editing delivery, and then I think what they're doing is plating about 500 cells per well and growing them up. Those were iPSCs. And so what happens is that you might have this 96-well plate where you have all these pools of edited cells, and you would basically grow them up a bit and cryopreserve half of them. So it's essentially like replica plating. You preserve about half of them, and then you interrogate the rest with ddPCR, and you identify the wells that contain a rare edit. And what you can do then is go back to that particular well that you've reserved, in the freezer and expand it. So you then take that well, and now you expand that across another 96 wells, and you repeat that. So you're enriching, you're saving half, you're testing half, and then you're enriching the pools that contain these rare edits. And so what's neat about ddPCR is, a lot of people, they might be like FACS sorting, they're sorting single cells and then growing them up. And that takes a lot of time and real estate, or some technician is just doing that. So with this approach, what you can be doing instead is screening the pools, figuring out where's my best, hit, what has the most, even if it's a very, very low level, it might be your best bet, and then focusing on that pool, expanding it, and so on. So that's the approach that the Conklin lab developed to enrich the pools for the edits. And the technology, the ddPCR, is what's enabling them to do that sort of screen, then clone, as opposed to clone, then screen.
Okay, that's all the questions we have from the audience. So thank you, everyone, for attending the Bio-Rad Droplet Digital PCR webinar today. If we didn't have a chance to get to your questions, someone from Bio-Rad will follow up with you directly in the next week or so. Be sure to check out the items in the resource list on your screen, including the slides. And please feel free to reach out to your local Bio-Rad Droplet Digital PCR specialist for additional information, or visit us online at biorad.com. Thanks, and have a great day.